Samson Prime Minister Award, and also he's a member of National Academy of Engineering in the U.S. and Science in the U.S. Before giving him a podium for his lecture, we will have a short ceremony for giving a certificate to Professor Jens Nielsen as a token of appreciation. So let me read the certificate. Presented to Professor Jens Nielsen, in general appreciation of your outstanding lectures at the 10th Annual KAIST CBE Global Distinguished Lectureship, October 26, 2020. So we will send this certificate to you by mail.
Thank you very much. Thank you.
Yeah, thank you. So, let me give him podium for his lecture regarding metabolic engineering. Professor Jens Nielsen, thanks so much.
Thank you.
Jens,
Yeah.
we forgot to take a photo. First, we wanted to take a photo in front of this podium, and then you will start. How about that?
Yeah, very good.
Yeah, thanks so much. Could you come up? Yeah, everybody come.
Jens, this is Sang-Yap.
Hello, Sang-Yap.
Yeah, I'm not going to be in the photo, but I'm looking at you now.
Oh, okay, good.
Many congratulations.
Yeah, thank you. Thank you.
A lot of people are listening to your talk.
I can see that, yeah.
About 130 people.
Yeah. But are there people in the auditorium also?
No, because of the COVID-19, the preparedness, they are all scattered. So in the auditorium, major professors who are available are there, while others are all joining from their desk through the Zoom.
Yeah.
YouTube channel as well. So many international people access to YouTube. So Professor Jens Nielsen.
So thank you very much for receiving this prestigious lectureship. I'm very honored. I'm sorry I could not be in visiting KAIST at this time. That would be fantastic. Hopefully that will be another time in the near future. But thank you very much for bestowing this award on me, and I hope to see all of you and meet and interact more in the future. Thank you. Okay, I think I will begin the presentation, and I hope my slides are all available. So again, thank you very much for that kind invitation to give a presentation in connection with this distinguished lectureship. So my first lecture here is about systems biology for metabolic engineering. The concept of metabolic engineering is probably familiar to many people, including in KAIST, which has a very active program in this space. But what it does, it enables cell factories to be used for production. Let me just see here what happened. I cannot go back. Oh, there we are. Okay, sorry about that. So somehow I suddenly got muted, but everyone hear me now, I assume. What metabolic engineering is enabling engineering of these cell factories so they can be used broadly for production of fuels and chemicals. And what is particularly interesting is that also, of course, along the time, we can further improve the performance of these cell factories and hereby improve the economics of the process, as well as we can refactor the engineering, the cell factory to produce other compounds that may be needed in the market. The challenge, and there's been many success stories already, but the challenge has generally been that it takes still a long time when going from what I call a proof of principle strain, that means a strain that has been engineered to produce the chemical of interest, until you get a strain that has performance in terms of titer rate and yield that can actually meet the requirement for industrial implementation. So traditionally, this has taken many years and been very costly in terms of the amount of person years that needs to be invested in order to develop this strain. So we really need novel technologies. We have seen some significant developments, particularly through genome editing tools, but we still need to have a more basic understanding of the cell factory before we can speed up this development. So this is where systems biology will come into play, and I will talk about it in my talk. Systems biology is also being now used widely, not only in this first step where you are developing the cell factory and making it more efficient, but actually also in the scale-up process, in the design of exactly what type of fermentation process are you going to use for the industrial scale production. So in my talk, I'll focus on yeast as a cell factory. This is something that we have done in my lab in the last almost 10 years, really narrowed in and looked at using yeast. There are many reasons for this. First of all, it has already been used widely as a cell factory. Here are some historical landmarks of this, of course, back traditionally on fermented drinks and leavened bread. Since several thousands before a common era. But also in more recent time, it's been, of course, used widely for production of bioethanol, various types of vaccines, but also various pharmaceutical products, and more lately also for chemical compounds that are used both as chemicals for example, for plastic production, but also fuels and antimalarial drugs. And the reason for the popularity of yeast is that it has many advantages, extremely well characterized. There's a lot of information. It's easy to engineer. It's very robust, so it's easy to scale up. And for these reasons, there's also a lot of focus on expanding further using yeast for production of various compounds. So over the years, we've been working on engineering yeast for production of many different compounds, some of them natural, for example, ethanol that is produced already, but improving the efficiency, to products that yeast don't naturally produce, but still making it such that they are produced at levels where we can get close to starting commercial production. And so these range from fatty acids, hydrocarbons, all the way, which are still in the kind of high volume, low value added space, down to more valuable products that can be nutraceuticals like resveratrol, antibiotics, and so on, all the way down to recombinant proteins, insulin, and hemoglobin. And along the way, we've also been worked on developing synthetic biology tools as well as systems biology tools that can enhance our engineering capabilities of yeast. So let me start by giving a couple of these examples before I turn to look into how we can use systems biology more actively going forward. So the first example is engineering yeast for production of fatty acids. We worked on this for several years. And initially what we did was we expressed a number of key enzymes, particularly ATP citrate lyase, that ensures provision of acetyl-CoA in the cytosol for yeast, together with also a fatty acid synthase from an oleogenous yeast in order to efficiently produce the fatty acids. And then we deleted a number of enzymes engaged in degradation of fatty acids, either through beta oxidation or also activation into CoA ester for further degradation. And also we took the free fatty acids, converted them onto alkanes and fatty alcohol. In this initial work, we were able to produce about 10 grams per liter in fed batch cultivations of free fatty acids. It turned out to be more difficult to convert the free fatty acids down to hydrocarbons and fatty alcohols, particularly the hydrocarbons, the alkanes. The enzymes are not expressing very well in yeast, so therefore we took a route of really focusing on just high-level production of free fatty acids, followed potentially by chemical hydrogenation of these free fatty acids. So in order to proceed on that, we further engineered the central carbon metabolism of yeast. So what we did was that we expressed a number of additional enzymes as well as also in the central carbon metabolism. And this resulted in very high level production of free fatty acids. And we were therefore wondering if we could actually cut away completely the production of ethanol, which is, of course, yeast is very good in producing that. And we did that by deleting the three enzymes coding catalyzing the first step, namely pyruvate decarboxylase and the conversion of pyruvate to acetaldehyde. And normally, if you delete all these three enzymes in yeast, it doesn't grow in excess glucose conditions because it doesn't have the ability of the Crabtree effect anymore. But actually, because we had installed this alternative route to produce free fatty acids, the yeast could grow, but it grew kind of slow. So what we did was that we used adaptive laboratory evolution, and then we actually managed to get it to grow faster. And we found causal mutation in pyruvate kinase that resulted in attenuation of activity of pyruvate kinase, and we found that that was then important in actually balancing flux through the glycolysis with the new pathway we had installed for efficient free fatty acid production. The resulting strain could produce more than 30 grams per liter, and we had a 30% of a maximum theoretical yield. And so actually when you run fermentations of this, you get this massive secretion of free fatty acids, so you get almost like a butter in the fermentation tank. We were also interested to produce medium chain fatty acids. There is quite some interest in that from the chemical industry. It can both be for direct use as solvents, but it could also be used, for example, in blend-in fuels. So if we could, for example, produce C8 that would and hydrogenated, it could be directly octane, but also C10 and C12 free fatty acids are of interest. And so what we did was that we looked at the fatty acid synthase, the endogenous one from Saccharomyces cerevisiae, and could see that in the catalytic chamber of this multifunctional enzyme, there's actually space for inserting an additional thioesterase activity. And when we did that, we could cleave off the growing carbon chain in the fatty acid synthase when they reach a carbon length of 8 or 10, or in some cases also 12 carbon atoms. And hereby we could produce these medium chain fatty acids. The challenge, though, we were facing is that these medium chain fatty acids are quite toxic for the cell. So we went on and particularly when we were looking into engineering further for higher level production, we observed toxicity. So we took an approach where we combined expression of a transporter at TPO1 and expressed that for secretion of these medium chain fatty acids. But we also used adaptive laboratory evolution in order to find mutations that generally improves the property of this strain. And hereby we could eventually improve both the secretion but also the growth and production of these medium chain fatty acids. We were also looking into production of longer chain fatty acids. These are relatively rare. They are produced by certain plant species, but it's generally difficult to source them. And they have interesting applications, for example, in cosmetics, also in shampoos and others. And particularly jojoba oil is of interest, and that is based on actually an ester of C22 carbon length fatty acids and fatty alcohol. So we looked into if we could produce this very long chain fatty acids, and we did that by combining expression of a mycobacteria fatty acid synthase that produces long chain fatty acids, and we combined that with expression of the overexpression of the endogenous elongation system of yeast, and hereby we could produce long chain fatty acid C22. We could also convert that into the fatty alcohol, which is docasenol, and then that could be used going forward. We were also interested to see if we could produce a novel class of compound, oleoethanolamide. These are actually naturally produced in the human body, and they play a role in actually controlling key fatty acid or lipid metabolism pathways. And so, for example, they play a very important role, for example, for treatment of dyslipidemia. And these compounds are quite complex to produce because they engage the phospholipid activity. And so, but through engineering of the phospholipid metabolism, we were able to really increase the production of OEA by more than 90-fold. And hereby we begin to get to levels where one could produce these compounds and begin to test them for potential clinical applications. We have also been looking into production of Cesco Terpenes. Cesco Terpenes is a very broad class of chemicals. Some of them have very nice smells, so they are actually used in the perfume industry as perfumes or fine fragrances. And some of these that are currently used are hard to source because they come from rare plant species. So the industry is interested to actually have a scalable production of these compounds. So we engineered yeast for high-level production of these Cesco Terpenes. That involves both engineering the central carbon metabolism as well as the delivering of the precursor. This is farnesyl pyrophosphate, and then also high-level expression of the Cesco Terpene synthases. And based on our work, Firmenich, our collaborator on that, a Swiss company, took this further, and they are currently now producing several of the fine fragrances that are sold on the market using yeast-based fermentation. We've also been interested in producing ornithine, that is a dietary supplement, but more interesting is also precursor amino acids for polyamines, for example, spermidine and spermine. These polyamines have a number of interesting properties. Actually, there has been clinical studies recently on spermidine for attenuation of development of Alzheimer's, but also of cardiovascular diseases. And that's simply because spermidine has been shown to be an inducer of autophagy and hereby regulates a key pathway in the human body. So what we did was we used a multi-engineering approach where we engineered both the supply of ornithine that is partly synthesized in the mitochondria of yeast, and we looked into various strategies and could then get high-level production of ornithine. We've taken this further now and also to produce spermidine, where we can also reach more than 2 grams per liter in fed batch cultures. So we've also looked into production of another compound that is of interest as an antioxidant, that's resveratrol. This can be produced in yeast by expression of resveratrol synthase together with also 2 heterologous enzymes that activates coumaric acid to coumaryl-CoA. And so through this pathway optimization as well as expression of heterologous enzymes, we could get high-level production. And this is also now a commercial process produced. And the compound is produced by Evolva and sold into the market for cosmetics and active ingredients in dietary supplements. Resveratrol belongs to this wider group of compounds called flavonoids. There's much interest in flavonoids. And so we were looking into see if we could have a general platform strain for production of high level of coumaric acid, and then coumaric acid to be further converted into various types of compounds. So what we did was we took a multi-engineering approach where we looked into efficient provision of the aromatics in yeast. This aromatics is in yeast, just like in other organisms, produced from phosphonyl pyruvate and erythrose 4-phosphate. And we used a phosphoketolase to elevate the level of erythrose 4-phosphate, and that turned out to be quite efficient to provision of the precursors for aromatics biosynthesis in yeast. Hereby, we could actually begin to get high-level productions of various compounds, various aromatics that are of interest in this area. We could produce high level of coumaric acid, but also could take this onwards to produce, as I said, some various interesting bioactives. So in following description of some of these cases here, I would like to return and say what are some of the real challenges that we are facing when we go through engineering cell factories. So generally what we are facing is that we want to produce this new product. This product is derived from a set of precursor metabolites in the central carbon metabolism. We also typically would need to balance ATP and as well as cofactors NADH and NADPH. And in order to do this, this is often our biggest challenge, is to balance the provision or efficient provision of the precursor metabolite as well as the ATP and cofactors NADH, NADPH balances, because they are involved in a large number of reactions. And so that's often the hardest challenge we have, this rewiring of central carbon metabolism in order to get to the high yields that are required for producing our product. And so that challenge, because it engages so many different reactions, is really something that is better approached using computer simulations. So we would like, of course, like in any other engineering discipline, to have computer-based design of our cell factory in the future. So for this, we are using Genome-Scale metabolic models. These models are comprehensive compilations of all the reactions that are occurring inside a cell. We are collecting information about the stoichiometry, the cofactor usage, and so on in the individual reactions. Which enzymes are catalyzing these reactions? It can be, in this case, as isoenzymes, 2 enzymes catalyzing the same reaction. It can also be protein complexes, where the enzyme is made up of several peptides. But in all cases, it gives us a link between the reactions, proteins, as well as the genes in the genome. And when we do this on a global level, we get a large metabolic network, but we also have a mathematical representation of cellular metabolism that we can use for model simulation using the concept of flux balance analysis. There's been a lot of work over the years on assembling such model for yeast, and recently we published the Yeast 8, which is the 8th iteration of a, you could say, community-based model. What we did was that we installed a version controlling of building this model in GitHub, so it can really be done in a community fashion, where if anyone discovers a mistake or a new reaction that needs to be added, it can be done. We can then evaluate that, annotate that, and it can go into the general model, so it can really be updated over time. In connection with building the Yeast Eight, we also annotated the number of additional reactions. So today we have the most comprehensive model with about close to 4,000 reactions linked to about 1,100 genes and more than 2,600 different metabolites. This is somewhat double counting because some of these metabolites are counted in their respectively compartments. For example, we count ATP, NADH, and so on in both mitochondria and cytosol, and in order to make sure that you can balance in these different compartments. Already now, the Yeast 8 has been further updated, and as I said, this is controlled by version controlling, and so whenever new information comes into the model, that is added, and I think we are currently at 8.4 in the yeast model. But we were also interested to take this further, and so besides this general metabolic network reconstruction, also look into some protein constraints application, enzyme constraint. I'll talk particularly about this. We've also recently looked into the temperature effect, for example, looking into the stability of enzymes at various temperatures. We also looked into collecting information about protein structures and so on. And I'll only give a few examples of some of these applications here. So, but one thing that is, of course, we were wondering was when we make a model for yeast or Saccharomyces cerevisiae, to be more specific, which strain are we actually talking about? So we were curious to see if there was actually variations across different strain species, and more than 1,000 different Saccharomyces cerevisiae genomes have been sequenced recently. And when we look into these genome sequences, we see that the pan genome saturates at about maybe 7,800 genes. Each yeast species has about 6,000 genes. The core genome is about close to 5,000, and the accessory genes is then about 2,800. And what is, of course, really interesting to know is how many of these accessory genes are actually part of metabolism. And it turns out that most metabolism is actually really in the core genome, core set of genes across these species here. There are a few peripheral genes that are responsible for metabolism. Most of this is related to utilization of various carbon sources. And so what we did was we began to compile all this information in what we call a pan yeast model. And so we just have a model that represents all the possible reactions that can exist or that are found in Saccharomyces cerevisiae. And then what we can do is that we can then, for each strain, we can simply take the gene content and then pull out from the pan yeast a specific model for that particular strain. So hereby we could generate easily models for 10,000 or 1,000 different yeast strains. And this is again also being updated, as I said, in the version controlling on the web. So, but one thing that the traditional genome scale metabolic models do not account for is actually the proteome constraint. And I often use this figure to illustrate that when the cell is operating, it can, of course, use various different metabolic pathways inside the cell, but these pathways don't come for free. Because for a pathway to operate, of course, you need to have the enzymes expressed, and the cell only has a certain constrained capacity to produce and express all these different enzymes. And so if we look at the protein content in yeast, it's a remarkable constant across many different growth conditions of about 45% of the yeast mass. And here you can see data from Quantitative Proteomics for yeast grown at two conditions: reference condition 30 degrees, chemostat culture, dilution rate 0.1, and also chemostat culture, dilution rate 0.1, but elevated temperature, in this case here 38 degrees. What happens is that actually yeast metabolism is shifting. You can see that allocations of proteome mass for the ribosomes is about the same because the yeast is growing at the same growth rate, but it starts to allocate more for glycolysis because it actually turns on fermentation, and less for oxidative phosphorylation. So you can see when you have to overexpress certain enzymes, it has a consequence on other enzymes, which then have to be expressed at a lower level. This actually has very important implications for, you could say, expression of heterologous pathways, but also rewiring metabolism in yeast, and I'll come back to that. So we incorporated this in our GECKO modeling framework, where we account for enzyme kinetics and cost. So we also look into what is the size of the proteins. So you could say if you have a pathway with very large enzymes with very low kcat, this has a very low efficiency compared with another pathway with small enzymes and very high kcats. And that information we can use to constrain the individual reactions, where we simply put an upper constraint on flux through the different reactions as kcat times the enzyme concentration. And remember again that this total sum of these enzyme concentrations here has an upper bound of this about 45% of the masses. So we collected kcats and mass information for all, at that time, about 800 enzymes in the GEM. And when we do that, we can actually begin to describe effects like the Crabtree effect, which a traditional flux balance analysis cannot do. And this is this shift in metabolism you get when yeast grows faster and faster. As at a certain stage, it simply has to turn on ethanol production, and you start to see that happening. You get a shift here in metabolism where you reduce the contribution of oxidative phosphorylation. I'll come back to that later. And you start to increase flux through glycolysis. You also see that by the specific glucose uptake rate that takes a kink here. It's increasing with growth rate here, but then at this stage, it's increasing at a higher rate. What we can also use the GECKO modeling concept for is that it can actually calculate the abundance of the individual enzymes in the network. And we can then compare that calculation with the enzyme usage, with the measured about the protein. So when we calculate that amount of enzyme used by the model and divide that by the actual measurements of the protein, we get an indication of what we call the enzyme usage. You could say that this is to some extent representing saturation level of the enzymes. We deliberately don't use that term because there could also be other factors playing in, for example, protein phosphorylation that could also affect, for example, the activity of the enzymes. And we calculated then the enzyme usage under 10 different conditions. These were actually 3 different types of stress at different elevation level, ethanol, temperature, and salt stress. And in all cases, as well as a control condition, and in all conditions, we can see that some enzymes have really close here to total usage, meaning that they are really used at full capacity. But it's a relatively small fraction, actually, of the enzymes in yeast. It is also varying depending on condition, which enzymes are fully used. But you see that a lot of the enzymes are used less. Actually, some are used extremely little. So you may wonder, why are these expressed at that level they are when they are used to this low extent? So this allows us actually now to begin to get a grip on enzyme usage at a group genome level. So let me shift on to actually illustration of use of this concept to get new insight into regulation of metabolism. And so what we were interested to do is to dig particularly into the central carbon metabolism of yeast. And so for this, we did an experiment, a very traditional or classical experiment where we grew yeast in different growth rates using chemostat cultures. And for each dilution rate or each growth rate, we sampled data for RNA-seq, quantitative RNA-seq, quantitative proteomics, phosphoproteomics, also metabolomics and flux quantification. And so all data were absolute quantitative, means we had information about copies per cell. So if we look first on the quantitative proteomics, we actually, there's something strikingly here. You can see again this map where we collected the data inside a square to really illustrate that you have this total constraint. You can see that you get more and more allocation of proteome mass to translation to the ribosomes. But one thing that really surprised us was that if you look at the central carbon metabolism, which to a large extent here is glycolysis, is actually having less allocation of protein mass. This is seen particularly if we look at a traditional plot like this. Here you can see the ribosomal system here, translation machinery, is linearly increasing with growth rate. So that seems really, when cells grow faster, they need more ribosomal activity. You can see that amino acid biosynthesis is increasing, maybe tending to bend off at higher growth rate. And then you can see also nucleotide biosynthesis is also increasing linearly with growth. Whereas when you look at lipid biosynthesis, that seems to be relatively constant, but then it's decreasing here at higher growth rate. We were first a little bit puzzled by that, but what is of course also happening is that when you go to high growth rate, due to the Crabtree effect, you actually get less mitochondria and therefore less inner phospholipids in the cell. Also, the cell size is actually increasing with growth rate, so therefore you have less cell membrane per mass. So that actually results in a lower lipid content in terms of phospholipids for faster growth rate. But what is really surprising, as you can see, is that the glycolysis allocation of protein mass for glycolysis is decreasing here, maybe reaching a plateau here for higher growth rate. And this, particularly in this regime here, is where you saw this rapid increase in glycolytic flux. So that's particularly remarkable that you don't see any increase here. So we looked in specifically to glycolysis. Here you can see quantification of the messenger RNA in proteins, phosphoproteins, and fluxes. You can clearly see both at messenger RNA level and protein level, you have decreased, except maybe for a few enzyme levels of the enzymes, whereas you have increasing flux through these enzymes. So we were thinking there could be various reasons for this. It could, of course, be because of the kinetics of the enzymes. So let's look at and we therefore took all the metabolomics data we had available and looked at if we could explain part of these changes in flux actually with increased saturation, for example, of the enzymes, or simply elevated levels of substrates for the individual reactions. And so we developed a simple log linearized kinetic model for each individual enzyme. And then we were looking if that could describe the change in flux over time. And you can see for some of the enzymes here, PFK, hexokinase, these are the upper enzymes here, we do see a tendency to that actually the changes in the metabolite concentration can explain the flux increase. However, what really is the major determinant of this is that we get a decrease in the phosphorylation of the enzymes with increasing growth rate. And so we are confident that this decrease in phosphorylation actually results in activation of the enzymes. So even though you measure a lot of protein, you have much of it inactivated due to phosphorylation. And so therefore you can activate that rapidly and then thereby increase the flux through glycolysis. We then calculated also the enzyme usage across, and we find that for most of the enzymes, there is an increase in enzyme usage. It's hard to see here, particularly for some of them, you can see they're kind of going up and down here. This is because when we have isoenzymes, the model will shift between using either one enzyme or the other, depending on simulation. But for most of them, you see this linearly increase in enzyme usage. So basically also meaning that when we increase flux and growth rate, we tend to saturate more of the enzymes in the cell. And if we look across and see where is this enzyme saturation, particularly or enzyme usage, really increasing linearly with growth, that is, for example, in amino acid activation of the tRNAs. So you clearly see that here. But you also see generally in amino acid metabolism here. Here's for the p-values for whether you have an increase. And here what you see, what you see in this axis here is the fraction of how many of the enzymes in that pathway actually had increased saturation. And you can see here that for some of the enzyme biosynthetic pathways, it's about 60 to 70% of all the enzymes in a given pathway that has this increased saturation across the board. So we were therefore interested to see what happens with the amino acid levels. And here I was very surprised to see that actually most of the levels of the amino acids decreases with growth rate. It's not a very strong significance here, but you clearly don't have an increasing trend, what we would expect it from these calculations. But it turns out that if we then, instead of looking at the amino acids, the final amino acids, we look at intermediates of amino acid biosynthesis, and they are shown, some of them here, you see that there you have increasing levels with increasing growth rates. And so this goes across for different biosynthesis of various amino acids. So this led us to generate this overall model for regulation based on this. That we have increasing level of many of the intermediates indicated here by red. We have increasing level of the enzymes in amino acid biosynthesis, so that allows to give a higher flux. Interesting enough, the final level of the amino acid is kept at a low level, maybe constant, but maybe even decreasing. That doesn't really matter because you can still load the tRNAs very efficiently, because the Km for the amino acids binding to the tRNAs is really, really low. So therefore, it's pretty insensitive to the concentration of the amino acids, but it's really determined by the tRNA levels. The advantage also of keeping the amino acid low means that you don't activate, you don't inhibit GCN4, which is very important for inducing expression of all the enzymes. And so this we could also see from our analysis, actually, that GCN4 is activated at higher growth rate. So this is an example of how we could get insight into global regulation, and this, of course, you can directly use for designing biosynthesis of amino acid biosynthesis, for example. We could do that across all pathways, where we could map out whether you have transcriptional regulation, that means you just overexpress the enzyme, or whether you have enzyme saturation or protein phosphorylation or allosteric regulation by the metabolites. And this we could do, as I said, for all the different pathways in the cell. So, but then let me move on and talk a little bit about what happens at nitrogen limitation. So, because these were experiments where we had carbon limitation in the chemostat culture. So what happens at nitrogen limitation, you begin to get really limitation in terms of synthesis of the ribosomes. And so there the cell actually starts to focus more on pure allocation of the ribosomal activity. And you can see here that you actually have that the ribosomal proteins, there are some that track with biomass. That means that basically they are, and here we're showing it more and more carbon limitation, or sorry, nitrogen limitation. Some are downregulated in the nitrogen limitation, and some track generally with the proteome here. What we observed was that at nitrogen limitation, you get actually a generally reduced proteome in the cell, and you get increased saturation. So this is illustrated here when we calculate this pathway usage based on the model simulations I showed before. We can see that at carbon limitation, you have maybe a relatively pathway usage of about less than 50%. But when you go to nitrogen limitation, you begin to saturate all the enzymes in the metabolism. And actually also what you do is that when you look at the ribosomal proteins, there are a number of iso compounds that the yeast can use, one or the other. It actually begins to control better the expression of all the compounds, so they are in a more correct stoichiometric relationship with each other. And so hereby it also saves in producing protein mass for translation. So let me end by talking a little bit about a global, where we could maybe draw some general conclusions about energy metabolism and how this proteome constraints really plays in. And so what we did here was that we expanded the modeling concept to look at both E. coli and Saccharomyces cerevisiae. Both organisms are characterized by having overflow metabolism. In the case of E. coli, that is in terms of acetate production. And what you can see is that what we calculated, we summed all the enzymes for, you could say, what we call a high yielding ATP pathway, that's glycolysis together with TCA cycle and respiration, compared with a low yield ATP pathway, which is typically the overflow, either ethanol or acetate production. And then we also summed pathways for precursor biosynthesis and biomass formation. And you can see down here a summary of some of these pathways here. You can see the high-yielding pathway of E. coli gives 23.5 ATPs. The protein cost is given here. And then we could calculate a protein efficiency, which is the ATP yield divided by the protein cost. And you can see that both for E. coli and yeast, even though the low-yielding pathway in terms of ATP gives much less ATP than the high-yielding one, in terms of protein efficiency, they're actually more efficient. So meaning that you get more ATP per protein mass allocated. And that's, of course, because these pathways are much shorter. They don't meet the requirement for all the protein mass in the TCA cycle and the oxidative phosphorylation. And so actually with these simple model simulations, we could describe the changes by simply writing overall stoichiometry for these pathways here. We could simulate the changes in overflow metabolism. Interesting enough, if you look at the protein allocation to energy metabolism, it's extremely conserved. So it's really a reallocation within those different pathways. You see this is based on quantitative proteomics for both E. coli and Cerevisiae at different glucose uptake rates here. But so this means, of course, that as you have less flux through the particular glycolysis and lower growth rates, it means that you have really less saturation. So you actually have less saturation. You reach an upper level of saturation of the pathway, and that's when you turn on then the overflow metabolism. This can also be used to begin to find strategies to improve growth, because what you can do is that you can either decrease the ATP demand for growth, or you could increase the proteome allocation for energy metabolism. And actually, we found examples from the literature where different approaches has been taken, typically using adaptive laboratory evolution. And so, for example, for increasing growth rate of E. coli, we have seen that there's example here where the proteome allocation has increased. So basically, the cell now allocates more protein mass for energy generation, or it simply decreases the ATP demand, typically by improving or reducing cost for maintenance. And similar, we found for a study in yeast, this was for high temperature growth. And finally, we've expanded this modeling concept to consider incorporation of metal ions in yeast. And this is quite important because metal ions are playing an important role in many key enzymes. And so now we can actually predict also the metal ions abundance in biomass. These are then simulated data versus measurements here. But more interesting, we can begin to look at what are the consequences of usage of iron in different pathways at different pathway fluxes, for example. And as well as what happens if we express complicated heterologous biosynthetic pathways that involves a lot of P450 enzymes. P450 requires iron. And so this, therefore, we took an example from a recent paper from Christina Smolke, where they expressed noscapine in yeast. It's a heterologous plant biosynthetic pathways with 4 or 5 P450s with relatively low Kcat values. And so therefore you need to actually have really significant additional iron content. And that has consequences on terms of central allocation of iron for other pathways, for example, for respiration. And therefore expression of this heterologous pathway can now begin to pinpoint exactly what elements of yeast metabolism you should engineer. So let me end with a summary of these implications of this proteome allocation for metabolic engineering. It can be visualized in this simple fashion here, that when we express a heterologous pathway, we basically allocate now a certain proteome mass for this heterologous pathway. And this has consequences because it results in a reduction of proteome allocation for central pathways that the cells still need. So what we need to be careful about is to look at the efficiency of these pathways. It can be that we should maybe overexpress key enzymes or maybe even downregulate enzymes that are already not very used, for example, because of very low saturation. And so hopefully I have demonstrated to you that yeast can be engineered to produce a wide range of chemical, but really a major challenge, as I discussed in metabolic engineering, is the rewiring of the central carbon metabolism. But the genome-scale models, particularly when we begin to incorporate proteome constraint, we can have very powerful models that can be used for cell factory design going forward. And so with that, let me acknowledge all my fantastic co-workers, postdocs and PhD students, and the funders, and you for your attention. Thank you very much.
Thank you so much, Professor Jansen Nielsen. Okay. So any questions from the student or floor or for international, audience, whoever? Any questions? Yeah, Professor Kim, Hyunwoo Kim.
Thanks so much for your talk, Professor Nielsen. Really enjoyed hearing your talk. So I get a feeling you've touched almost all aspects of yeast. Having said that, do you have any particular objective that you have in mind in the coming years? That's my first question. And the second question is, so now that everyone talks about AI in all technical domains, do you happen to have any particular view towards AI for metabolic engineering?
Yeah, thank you for the great questions. I think in terms of focus on metabolic engineering for yeast, right now we do have a focus on natural products that can be used for bioactives in general applications. It can be nutraceuticals and others. But also there's a lot of interest now in of course, production of plant-based proteins, fats, and so on. And if we can begin to do that, for example, using even biomass as a feedstock, we will begin to be able to produce food using biomass as a feedstock. So I think that's an extremely fascinating area, still extremely challenging to go in terms of cost, but that could allow to begin to go in a direction of more healthy food production. So that's what I'm very excited about. In terms of your question about AI use, so there is of course a lot of interest in using AI, and actually we have used AI. I didn't talk about it here, but that was in connection with design, or you could say finding, developing a yeast model for description of yeast growth at different temperatures. So in order to do that, we needed to collect information about the catalytic efficiency of the enzymes at different temperatures. And there is limited information. So we could find that for certain key enzymes and so on. And so what we actually used AI for was for basically traditional regression analysis, but using AI to find the parameters for all these enzymes in yeast. And then we used also stochastic evaluation for scoring of these different parameters and so on. So we have a paper in the pipeline on this. And so I think this is where AI will begin to play a very important role, and particularly when you go to organisms that are maybe less characterized than Saccharomyces cerevisiae. Let's say we want to have information about the kcat values. How could we do that? Could we begin to use genomic information, structural information, and so on to predict kcat values across? So that's where I think it would become extremely useful.
Okay, any other questions from faculty or student? Yeah, any comment on this?
Thanks a lot for the answer. Just out of curiosity, in your research army, I should put it that way, do you happen to have a number as to the percentage of people who are dedicated to computational biology?
Sorry, can you please repeat?
Oh yeah, the percentage of people in your research group who are dedicated to computational biology. Do you happen to have that one? Yeah.
Yeah. So this is about 50-50, I would say, right now. Yes. And that's also what I'm going to talk about. And if we look at the yeast work, it's probably more than, you know, maybe 70-30%, 30 on computational. But as what I'll talk about on Wednesday is about studying on human metabolism, and that's all computational.
So any other comments or questions? I would like to have from students.
Can I have a question?
Sure, go ahead.
So first, thank you for giving a nice lecture for us. And I wonder, in your lab, do you have any plan to expand your in silico models to wholesale models, like for E. coli wholesale models?
Yeah, that's a very good question also. We do have a project, and we're almost done with the model where we have expanded with what you could say wholesale modeling, because what we did there was that it's similar to what Bernard Paulson called the ME models. We use another term. Well, basically what we model and quantify the description of production of all the proteins, including ribosomal protein, chaperones, and so on, including allocation them to different compartments and so on. So we actually begin to account for most of all the cellular processes. We still don't have transcriptional regulation. We still don't have, for example, DNA replication and so on. But besides that, practically all other reactions are incorporated. And that looks fascinating. We can actually get a lot of insight into what are the key constraints of the cells. And we can also describe a number of basic biological phenomena. I think, and I decided not to talk about it here because I think actually that type of model does not provide really many advantages compared with the more simple enzyme constraint models that I was talking about here for metabolic engineering. What these wholesale models, as we could call them, are valuable for is actually getting new insight into biology and asking deep biological questions about how are different processes interacting and so on. They're quite complicated to simulate also. So again, also, I would really, for metabolic engineering design, prefer to have simpler type models that are still capturing key mechanisms. And here, as I said, these enzyme constraint models are actually doing a very good job.
Thank you.
Thanks. For anyone who wants to give a question in terms of written statement, you can do in chatting, okay? Any other questions or comments?
Well, this is Sang-Yap. While students are maybe preparing their questions, let me ask you a quick question on protein production, because Saccharomyces cerevisiae has been a workhorse for recombinant protein production some years back, and of course it has been taken over by other host cell systems. But you have studied such a nice work on the Km value of amino acids against tRNA, etc. Have you tried overexpressing tRNA synthetase to overexpress recombinant proteins?
That's a very good question. So no, we have not done that. But actually, I don't think that that we do have a limitation in that. I mean, normally what you would do for recombinant protein production, you will see, of course, a competition against growth. So you will, if you high level express for example, even insulin, which is a small peptide, you will see a penalty on growth because you will begin to compete in the ribosomal system of expression of the heterologous enzyme or protein. But normally that is not a big problem. You would go either in fed batch mode and then you will turn on the expression, or you would use chemostat cultures, as is actually often done for recombinant protein production by Cerevisiae. And then you will just go to a slower growth rate regime, and then you can have the allocation of the ribosomes for production of that. And that goes even also for the tRNAs. I don't think that we have seen any indication that that's limiting. What is often limiting is the protein folding, the stress that causes, and further downstream protein glycosylation and stuff like that. So that's normally where you see limitation in recombinant protein production in Saccharomyces cerevisiae.
Okay, thank you, Jens.
So any other comments or questions from students? Or you can put it through chatting window. Comments? So I have a question for you. So even though I'm not biotechnology area, how can you upgrade your hydrocarbon into the fuel, especially when you use the hydrocarbon, which was from microalgae, I think. Then you upgrade it to fuel level. Then you have a technology transfer to Total in France. So how can you reduce your oxygen level in the hydrocarbon?
Yeah. Also a very good question. I mean, and today I think we are seeing dynamics playing out that is very hard to say where that will end. If we go back about 8-10 years ago, the fuel industry, including all the big players, they were all looking into bio programs and starting bio programs, and with the objective of beginning to have bio-based production of fuels. They were doing that predominantly, I think, out from a objective of potential future scarcity of fuels, but also maybe expecting governmental mandates and so on coming in the future. And a general belief that, you know, if enough resources was allocated into engineering microorganisms for fuel production, you will be able to produce these at a low cost and thereby compete with oil production. Then about 5 years ago, it started to change dramatically. I mean, most of them have left this space. But then in more recent years, you start now to see again demands. Now it's coming really from the political side, where many politicians around the world are beginning to put demands. For example, Sweden has now put a demand for blending on biofuels into jets, for example, and so on. And so you will begin to get other mechanisms into play where someone have to deliver these compounds, and not, you can say relatively independent about what they cost. And so that would probably result in renewed interest in this. And now let me get you to your question about how you can do it. Well, I think we actually did some calculations on if we were going to use Saccharomyces cerevisiae to produce hydrocarbons, let's say alkanes or alkenes, could it be compatible? I mean, and it's hard to say, you know, if you're going to compete with the oil industry because the oil price is fluctuating dramatically over time. But what we could clearly see was that we could produce, in terms of energy content, we can produce hydrocarbons at a cost similar to the ones for ethanol. So in other words, you know, if ethanol is a feasible product, then some of these new ones could also be. How would you do that? Today, I think you would, and that's actually how many of the biofuels are coming for jets today. That is about taking fatty acids from either food or forestry or other residuals and hydrogenate them, and then blending them into diesel. So I think you could do the same microbial production of fats, and then use them for hydrogenation, and that would probably be the approach going forward. And so I think we will see, it will be more expensive than traditional fuels from oil. It's super hard to compete with, particularly in the oil prices is extremely low now. But I think we will begin to see some developments in that direction also.
Okay, thank you. Here is some questions pop up from chatting window. Jens, can you see the question from the chatting window?
I can take the first questions and about how we can control the ATP demands. Very good question. It's very tough, but it turns out that actually, and this was examples from this E. coli adaptive laboratory evolution, that if you do select for increased growth rate, you actually begin to get a rewiring. And typically what it results in, it can be several different things, but it can, for example, be that you change the amount of ATP allocated for maintenance. And you say, how can the cells do that? Well, probably because the cell has evolved to allocate a certain amount of ATP for example, some of these growth-associated maintenance processes. But it doesn't need all of them. It just has evolved to have all this in place, because if it suddenly gets into stress conditions, so if you kind of adapt it to grow at certain conditions, you can shrink that expenditure. So that would be one approach to do. And but otherwise, this is probably one of the only examples I could think of. But that, because it is very hard to change ATP demand in the cell.
And second question from Dongshu Yang.
Yeah. So yeah. So this goes a little bit about the rewiring of fluxes in connection with natural product synthesis. So a very good question also. I mean, often some of these bioactive, we don't need to rewire, that much, because I mean, they're normally higher value added compounds. And it is exactly as indicated here. It is the bigger challenge to express the enzymes of the pathway in the organism that you're looking at. It could be E. coli or yeast for that matter. Many of them are maybe non-soluble, or they are missing cofactors that are not naturally produced by the cell factory. And so often, I would say for production of natural products, and particularly complicated plant biosynthetic pathways, it is often about assembling the pathway, focusing on having activity of the individual enzymes. When you have that in place, it can be that you may have a limitation in provision of, for example, as I mentioned, the example of the for coumaric acid production. You can do a few simple tricks in central carbon metabolism to deliver more of that, but often it's really more kinetic demands or kinetic challenges and enzyme expression demands. Let's look at the last one.
Yeah.
Yeah, that's also about the successful commercialization of natural products. So again, this depends, because natural products is of course many different things. You could go all the range from Resveratrol, for example, which you can extract traditionally extracted from plants. But even in those cases, we looked at, it still sells for in the order of a little bit depending on quality, maybe from $500 to maybe $1,500 per kilogram. So, it's completely different price than fuels and commodity chemicals. And so that's what you need to target. And of course, you want to make sure you can make money. So you should try to be able to produce it maybe at $100 per kilogram or maybe even lower. And then you have a good margin there. But then if you take another natural product, it can be, some of the things that Christina Smolka is working on, producing of opioids and opioid analogs, stuff like that. There you come into maybe, price ranges of $1,000 or several $1,000s per kilogram, maybe even 10,000s and so on. So there, the demand for an efficient process is of course going even further down, and you don't need that tighter rate and yield. So it depends on what type of product we're talking about. But I would say for most, and that's why there's a lot of interest in trying to identify compounds that are maybe in the range of $100 to $500 per kilogram, because that's a kind of a good target that you can develop a cell factory with reasonable efforts. Yeah. Did that answer the questions? I hope it did.
I think so. Thank you. Well, let's stop here. And then basically, if you have a question, basically you can prepare the question for Wednesday talk. So yes, we are more than looking forward to your Wednesday talking. So basically, let's stop here. Let's thanks again to the speaker. speaker for his comprehensive and excellent talk.
Thank you very much.
See you on Wednesday.
Yeah, see you on Wednesday.
Yeah, thanks so much.
Thank you very much. Bye-bye.
Bye.